This video abstract is AI generated. In heterogeneous catalysis, the active metal is often treated as the main reaction site, while the support is viewed as a passive scaffold. In this work, we asked a broader question: how do substrate-support interactions influence hydrogenation on PD/CeO2 nanorods? To address this, we examined a library of phenolic compounds including phenol, anisole hydroxyphenols, and methoxyphenols. Our goal was to understand how the number and position of oxygen-containing substituents affect adsorption and catalytic conversion. We used a simplified surface contrast NMR approach based on carbon-13 transverse relaxation. For each substrate, we measured the carbon R2 values in the absence and presence of nanoparticles. The difference, delta R2, provides a practical readout of how strongly a substrate interacts with the nanoparticle surface. These delta R2 patterns can be interpreted in terms of distinct binding modes. The first is hydrogen bonding to CeO2 through the phenolic OH group. The second is flat adsorption on PD, corresponding to a rigid substrate PD adduct. The third is binding at an oxygen vacancy on CeO, where the phenol is deprotonated and the oxygen inserts into the vacancy, allowing anisotropic rotation about the OC axis. Together, these modes explain how different adsorption geometries generate different relaxation fingerprints. Using this framework, we found that hydroxyphenols bind CeO2 more efficiently than phenol or methoxyphenols. This trend reflects stronger hydrogen bonding to the support through phenolic OH groups. In general, increasing the number of phenolic hydroxyl groups increases productive substrate support interactions, but not all adsorption is productive. Compounds with an oxygen substituent at the 2 position, such as 2-hydroxyphenol and 2-methoxyphenol, form unusually stable interactions with PD sites through the oxygen-containing substituent. These strong PD adducts correlate with lower catalytic conversion, indicating that overly stable substrate PD binding can inhibit hydrogenation. Overall, our results show that weak substrate CeO2 interactions promote catalysis, whereas overly stable substrate PD adducts reduce it. This study demonstrates that carbon-13 delta R2 measurements provide a fast and informative NMR screen for nanoparticle adsorption, and it highlights how support interactions can play a central mechanistic role in heterogeneous catalysis. To know more, read our work online.